Hello everybody, welcome to Ms. Maguire online video lecture, and today you will be introduced to stem cells. So let's go ahead and begin. What is a stem cell? Stem cells are undifferentiated cells that have huge capacity to self-renew. All cells in the body come from stem cells. So if you look at the diagram over here, here's a single cell that can replicate itself, or it can differentiate into many cells, other cells, right? Like your blood cells, your bone cells, cartilage, skin, and so on. Actually, at the beginning, during embryonic development, all your cells come from stem cells. Stem cells have not any function, and are not yet specialized. They can differentiate into more than one cell type. So no functions yet, can differentiate. Stem cells have ability to divide for indefinite periods. They should differentiate into appropriate cells type when transplanted to damaged recipients. So those are characteristic of stem cells. So a unique characteristic of stem cells can regenerate. They have unlimited self-renewal through cell division. That means that stem cells can divide and produce more stem cells, and this renewal is unlimited. This is unlike muscle cells, blood, or nerve cells, which do not normally replicate themselves. Stem cells can specialize under certain physiological or experimental conditions. Stem cells can become cells with a specific function, such as beating cells of the heart muscle or insulin-producing cells of the pancreas, right? So those are very important characteristics. Ability to regenerate or produce more stem cells, and its unlimited renewal ability, and specialization. That means that stem cells can become different cells of your body. So differentiation is when unspecialized stem cells divide. It will divide, and it will give rise to specialized or differentiated cells in response to external and internal chemical signals. Internal signals turn on specific genes causing different differential gene expression, and external signals include chemicals that secreted by other cells, such as growth factor, cytokines, and physical contact with neighboring cells. Right, so we already mentioned that function and characteristic of stem cells is differentiation, right? Now, so how cells differentiate? How cells know that they need to be a bone cell or a nerve cell or muscle cell? Well, they will respond to signals, either internal or external or both. Like internal signals, for example, if we send the signal to turn off a gene for production of collagen, for example, then the cell will not become your fibroblast. If we turn on a signal to produce insulin, then a cell will produce insulin, so it can be your pancreatic cells. Right? Those are internal signal, and external signal we will discuss in this chapter further, right? But it's going to be chemicals that will guide the differentiation of the stem cells into specific lineage. Okay, so a life story. We all start with two cells, egg and sperm, or ovum and sperm. Then cells, sperm and an egg combine, we call it fertilization, and we form the first cell called zygote, and then zygote divides. Okay, so I need back to my mouth. Okay, so and we have division, we form 2 cells, we form 4 cells, we have ball of cells, and then cells start first differentiation. And from those cells, the entire organism is formed and born, and you became adult, right? So here's a stem cell. It can self-renew, so it can copy itself and make another stem cells, and it can differentiate. Differentiation means specialization. Specialized cells can be formed from stem cells, such as muscle cell or nerve cells. So when stem cells copy themselves, they produce identical stem cells. When they differentiate, they produce specialized cells. Right? So why do we need those stem cells self-renew and differentiate? So here we start with one stem cells. So it divides, produce another stem cell, and differentiated cell, the cells that became maybe your neuron. It doesn't need to happen during the same division. Stem cells can divide and produce 2 stem cells, then they can divide and produce 2 specialized cells. But that's our first division. And then, so we have again stem cell that will divide, produce another stem cells and specialized cells. Divide, produce another stem cells and specialized cells. So at the end, as seen in this example, we started with 1 stem cell. We end up with 1 stem cell. So self-renewal maintains the stem cell pool. So we still have it, right? We didn't lose it in the process of division. And we end up in this example with 4 specialized cells. So differentiation replaces dead or damaged cells throughout your life. Stem cells can be classified into by two main criteria: by differentiation potential, totipotent, pluripotent, multipotent, and by source, where these stem cells come from. We have embryonic stem cells, adult stem cells, and induced pluripotent stem cells. Okay, so this is a classification based on potency. So totipotent, those are embryonic blastomere. So that's early embryon. It's from fertilization up to 8 cells stage. Totipotent stem cells give complete organism, including even extra embryonic tissue. So if we have totipotent stem cells, they are found during early embryonic development, right up to 8 cells stage. And those cells can differentiate in all the tissues and organs of your body. Pluripotent, also from embryonic tissue, but later, not when we have 8 cells, but when we have enough cells that will differentiate into primary or embryonic tissue layers. So when cells in your body differentiate already into primary tissue, ectoderm, endoderm, and mesoderm, those type of embryonic cells are pluripotent. And we have 2 types of pluripotent cells, EC and EG, embryonic stem cells and embryonic germ cells. So we will talk about this type of cells in a couple minutes. Then multipotent. They differentiate into more than 1 type of specialized cells. So example would be hematopoietic stem cells that differentiate into all cells found in your blood, right? Your blood cells. And progenitor cells. They produce terminally differentiated or specialized cells. So you see the potency, the ability to differentiate decreases from totipotent to multipotent and then to progenitor cells. All right, so here's our hierarchy of stem cells. Here's our totipotent cell, a very early embryonic up to 8-cell phase. This arrow shows us that this cell can renew itself. So this cell can make more stem cells. Then when those early cells differentiate into pluripotent, they can still renew themselves and they can produce many other type cells and tissues. So this one make a blood stem cells, tissue-specific adult stem cells, and some other cells. So those cells now here are multipotent. Like blood stem cells or hematopoietic stem cells can differentiate into red blood cells and white blood cells. Tissue-specific adult stem cells differentiate into muscles, nerve, epithelial tissue of your intestine, or other tissues. And multipotent cells over here, they don't renew themselves, right? So we have renewal in totipotent and pluripotent cells. Now, classification based on where they're found, where stem cells are found. So before we discussed classification of stem cells based on their potency, right? So how many tissues they can form, into how many cells they can differentiate. Now we're gonna classify them based on their source, where they found. We have embryonic stem cells, right? So here's our early embryon called blastocyst, and this area called inner cell mass. So that's our embryonic stem cells. We have induced pluripotent stem cells, or IPS. Those are cells of adult. For example, over here, there is a fibroblast from skin. Then different chemicals are applied, and we can reverse those specialized cells back to pluripotent stem cells. So we can reverse them back to pluripotent. So that's called induced pluripotent stem and adult stem cells. In this example is a muscle stem cell tissue. And right, so they are still undifferentiated and still can differentiate into more than one type of tissues and cells. Right, so here's our life story again. We start with egg and sperm, fertilization, zygote and zygote divide. And when we get to this point, when we have so that's a ball of cells here, and then we form what called blastocyst, and blastocyst have this inner mass, and that's where this is still early embryo, and this is where we find embryonic stem cells. Then we have a tissue or adult stem cells over here found in a fetus, in a baby, and throughout life. And induced pluripotent stem cells comes from a tissue stem cells, not even tissue stem cells, from a regular differentiated tissue stem cells that can be reversed back to their pluripotent form. Oops. Okay, so embryonic stem cells come from a 5 to 6 day old embryo. They have the ability to form virtually any type of cell found in the human body. And embryonic germ cells, so remember, embryonic stem cells, we have two types, embryonic stem cells and embryonic germ cells. So embryonic stem cells from 5 to 6 days old embryo and can be differentiated into all cells found in the human body. Embryonic germ cells derived from a part of a human embryo or fetus that will ultimately produce eggs or sperm. So embryonic germ cells, those cells that will produce gametes or eggs and sperm in human. So embryonic stem cells, where we find them? We find them in a blastocyst during early development in the inner cell mass. Then embryonic stem cells can be taken from the inner cell mass. They can be cultured in a lab to grow more cells within fluid with nutrients. Embryonic stem cells are derived from embryos that develop from eggs that have been fertilized in vitro only. So we do not take these embryonic cells that is inside a woman's body, right? Only from embryos that develops from eggs that fertilize in vitro. In vitro means outside of human body. Embryonic stem cells are never derived from eggs fertilized inside a woman's body, or at least in the United States. The embryos from which human embryonic stem cells are derived are typically 4 to 5 days old and are hollow microscopic balls of cells called blastocysts. So yeah, we're talking about embryo stem cells, right? And we said, well, where can we get them? Where those cells are? Because those are really very important cells in our health and our well-being. Well, we have embryonic stem cells, but just remember, we never use embryo inside a woman's body. We use embryo that was created in vitro. Right, so here's stages of embryogenesis, just to remind you. 1 day, fertilized egg. 2 days, 2-cell embryo. Day 3 to 4, multi-cell embryo. So this is our totipotent cells. And then when they form blastocyst, day 5 to 6, those are pluripotent. And blastocysts start a tissue differentiation in day 11 and 14. Now, when we form first tissue, ectoderm, endoderm, and mesoderm, then those cells now pluripotent. So they still differentiate into almost all cells of your body, but not all of them. A totipotent do. Totipotent differentiate into all cells and tissues. Oh. So this is how in vitro we grow embryo, and we can use the embryo stem cells. So here we have intracytoplasmic sperm injection. So that's an egg, and we have a needle in a special syringe that's gonna bring a sperm inside. So sperm will fertilize the egg. Here we have fertilized egg. Then 2 days embryo, so it starts dividing. So now we can see 2 cells, 4 cells embryo, 8 cells embryo. Then the ball of cells called morula. All right, so this is our totipotent, this one, totipotent, and then we have our inner cell mass. When we start differentiation into primary embryonic tissues. All right, so that's how embryonic stem cells are collected in labs. Right, so here now it's a diagram. We have, again, egg fertilized by sperm. We have morula, we have blastocyst, and in day 5 or 7, isolation, and we can isolate this inner cell mass and cultivate it, or we can in week 5 to 7 we can collect the cells from the organs that will be developed into ovaries and testes, cultivate them, and we get embryonic gonadal cells. So embryonic stem cells and embryonic gonadal cells. So I want to go back to show you where we cover this process right here. So when we're talking about pluripotent cells, we have embryonic stem cells. I'm sorry, not gonadal, and embryonic germ cells, gonadal. Gonads are your eggs and, I'm sorry, your ovaries and testes, but it's called embryonic germ cells. So right here, where we look at it, so if we collected day 5 to 7 embryonic stem cells, collect day 5, week 5 through 7, then it's going to be embryonic germ cells that collected from areas that are going to be gonads, testes and ovaries. Right. Okay. Why embryonic stem cells so special? Because they pluripotent stem cells. They can differentiate into 3 main tissue, ectoderm, mesoderm, and endoderm, and they can form more than 200 types of cells. Okay, so embryonic stem cells, what they do, they differentiate into all possible types of specialized cells, and they are pluripotent. Advantages of using embryonic stem cells are flexible. They appear to have the potential to make any cell. Immortal, one embryonic stem cells line can potentially provide endless supply of cells with defined characteristics and availability. Embryos from in vitro fertilization clinics, so we can easily get the embryonic stem cells. So that's advantages. We do have disadvantages. Difficult to differentiate uniformly and homogeneously into a target tissue. So if you have a bunch of cells, sometimes some of them differentiate, some don't, so it has some difficulties. They are immunogenic. Embryonic stem cells from a random embryo donor are likely to be rejected after transplantation. So if we have a patient that need organ transplant and we transplant organ from embryo, embryo, those cells are not this human cells, so it can be rejected. They also tumorigenic. Tumorigenic means they're capable of forming tumors or promoting tumor formation. And of course, another disadvantage is disruption of developing human life. So we just mentioned that, yes, we use embryonic stem cells that were formed in laboratory, right? In vitro, but still, if you have egg fertilized by sperm, that potential human life. So some people, or many people, I would say, have some problems about it, right? And they're not sure if this is what we should do. So that's a disadvantages. Okay, so embryonic stem cells have other challenges. So they go under condition A, they can become skin cells, or another condition, they can be neurons, blood cells, or liver cells. But what exactly induced their changes into precise type, right? So scientists around the world are trying to understand how and why embryonic stem cells produce skin, blood, nerve, or any other particular kind of specialized cells. What controls the process so that the stem cells make the right amount of each cell type at the right time? The big challenge for scientists is to learn how to control these fascinating cells. If we could force embryonic stem cells to make whatever kind of cells we want, then we would have a powerful tool for developing treatments for disease. For example, perhaps we could grow new insulin-producing cells to transplant into a patient with diabetes. But there is a great deal to learn before such therapies can be developed. Scientists also want to use stem cells to understand how disease develops and test drugs in the laboratory. So yes, we do have some challenges. We don't fully understand how stem cells, embryonic cells, stem cells are induced, and how can we use them without triggering immune response or development of a tumor. Okay, another type of stem cells based on location are adult stem cells. Adult stem cells is an undifferentiated or partially differentiated cells found in tissues and organs of the organism that is born already. They can self-renew and differentiate to become most or all of the specialized cell types with their specific tissue lineage. So that means if you, and we will, I will give you examples of adult stem cells. So if you take hematopoietic stem cells, hematopoietic adult stem cells can only differentiate into blood cells. They cannot differentiate into muscle cells, right? That's why it says within their specific tissue lineage, right? Adult stem cells maintain self-population. That means those stem cells, they will divide and produce more stem cells. They can help you heal and play a role in aging. Okay, so where are they found? Surface of the eye, in your brain, in your skin, breast, intestine, testicles, muscles, bone marrow, right? What can they do? They can differentiate, but they only specialized types of blood cells. If you, for example, if you take a blood stem cells or hematopoietic stem cells, you will find them in the bone marrow. They will differentiate, but they only differentiate within their specific lineage. So they will create blood cells, red blood cells, white blood cells, and platelets. They are multipotent. They are not pluripotent, and of course, they're not totipotent. Adult stem cells and progenitor cells reside throughout your body. These stem cells reside in a specific area of each tissue called stem cell niche, right? So they found in a specific area. This niche is a particular microenvironment that fosters the growth of resident stem cells. Mutations in cells, signals they receive, and changes in microenvironment can activate a stem cell. So a stem cell niche. So we said that stem cells are located within their specific environment inside your organs. So the stem cell niche is a major concept in stem cell biology. Understanding the microenvironment around stem cells is important, and understanding stem cells themselves. The microenvironment regulates the behavior of stem cells and thus can teach us how to control stem cells in future and in culture. The niche can act on the stem cells by various mechanisms. So as we said, let's go back. Now, stem cells reside in a specific area of each tissue called stem cell niche. Now, we need to understand what this environment surrounding stem cells is and what it does to a stem cells. And actually, this environment can activate stem cells by direct contact between stem cells and niche cells, by soluble factors released by niche that travel to stem cells, and by intermediate cells that communicate between the niche and the stem cells. Scientists still working to understand exactly how niches work, and more is known about the niches of some kind of stem cells than others. So here our niche for stem cells. It's microenvironment around stem cells that provide supports and signals regulating self-renewal and differentiation. So, sorry, what did I do? Somehow I exit my presentation. Sorry for that. Okay, so I just wanted to pick up my pen and exited my presentation. So here's the stem cells, and here's another cells within the organs that we call niche. Right, so now let's see how stem cells niche can affect stem cells by direct contact, by soluble factors, so they secrete factors that will activate stem cells and kind of give it instruction what type of cells it need to, what type of cell it need to differentiate into, or through the intermediate cell, right? Types of adult stem cells. So examples, right? So we already talked about embryonic stem cells. We can find them in early embryo, and we grow those embryo in laboratory. Those are pluripotent. Adult stem cells, they are multipotent. Then they can differentiate into cells within their own lineage. So here's an example of adult stem cells, hematopoietic stem cells that differentiate into cells of blood and immune system. Mesenchymal stem cells can differentiate into bone cells, cartilage, fat, muscle, tendon, ligament. Neural stem cells that can differentiate into neurons and supporting cells called glial cells, and epithelial stem cells that will differentiate into cells of your skin and lining of internal organs. Okay, so here are the principles of renewal in tissues. Now we have a stem cells, and it can self-renew, divide, and have high potency. Those are pretty rare cells when we're talking about adult stem cells. They can be committed into progenitor cells. So they will further differentiate into committed progenitor. Then they are transient amplifying cells, multipotent, divide rapidly, and they cannot self-renew. So remember, when we have a stem cells that totipotent and pluripotent, they will renew themselves, so they can make more stem cells. So that's what this arrow shows you. I don't know what happened with my, let me exit for a minute. My pen doesn't want to work properly. So I'm gonna go back and I pick up my pen again. Hopefully it will work better this time. Now, when they changed into, oh, that better, into progenitor cells, still stem cells, right? But multipotent, and they cannot renew themselves. Oh, maybe we cannot call them stem cells. If they cannot renew themselves, they're not stem cells anymore. They just progenitor cells. And then progenitor cells, they will divide into specialized cells. They will perform cellular function, and it will be no division. Specialized cells, most specialized cells do not divide, right? So here's our hematopoietic stem cells. They give rise to all blood cell types. They will differentiate into myeloid and lymphoid lineage. Myeloid will then produce white blood cells, red blood cells, platelets, right? Many types of white blood cells. Lymphoid will produce special white blood cells called T cells, B cells, and natural killer cells. Right, so next slide shows us hematopoietic stem cells, right? So they found in the bone marrow, right? They will be, they become the committed progenitor cells, and either gonna be lymphoid or myeloid lineage. And here we make white blood cells, right? If it's lymphoid, and if it's a myeloid progenitor, then we make also white blood cells, just a different type, like neutrophil, eosinophil, basophil, macrophages, but also red blood cells and platelets. Right, but keep in mind that hematopoietic adult stem cells can only differentiate into blood cells, right? So it's a limited ability to make cells and tissue. They are multipotent, but not pluripotent, right? Another example are mesenchymal stem cells. These stem cells differentiate into cartilage, muscle cell, fat, tendons, and ligaments, and connective tissue, including osteoblasts that found in skin, for example. These cells are located throughout the body. They found in bone marrow, in the fat, in the spinal cord, blood, and they are easiest to isolate. So here's our mesenchymal stem cells, right? For example, here found in the bone marrow can renew themselves. Then they form committed progenitors, and these committed progenitors will give rise of osteoblasts, chondrocytes, or adipocytes to a specialized cells. Yeah, again, for some reason, cannot make circles. Neural stem cells, they're located in subventricular zone lining the lateral ventricles. And if you didn't take anatomy class, maybe you don't know what lateral ventricles are. So I'm just briefly going to tell you that inside your brain, you have spaces filled with cerebrospinal fluid, and we call them ventricles. And two of those ventricles are lateral ventricles. And in those lateral ventricles, you will find neural stem cells, where they give rise to newly born neurons that migrate to the olfactory bulb via the rostral migratory stream. So they form inside the ventricles, and then they migrate into olfactory bulb, that is structure you used to smell. Olfaction means sense of smell. They also located in subgranular zone in the hippocampus, and hippocampus located in your cerebrum, right? So there's the largest part of your brain, and it's underneath the cerebral cortex. There is a area called hippocampus, and that's believed to be important in the memory. So we have stem cells in this area as well. Neural stem cells, also called neural precursor cells, give rise only neurons, oligodendrocytes, astrocytes, and those are all cells of nervous tissue. So neural stem cells cannot give rise to your bones or muscles. So again, they are multipotent. So here the diagram shows us neural stem cells found in the brain. It will be differentiated into committed progenitors, and they will produce neurons, interneurons, oligodendrocytes, astrocytes. Those will be specialized cells. Epithelial stem cells give rise to epithelial cells, which constitute 60% of the differentiated cells in the body. So 60% of your cells are epithelial cells. They are responsible for covering the internal and external surfaces, such as your intestinal lining, such as your skin, including the lining of vessels, glands, and other cavities. Epithelial stem cells are also found in a bulge region of a hair follicle. So I'll show you where they found. So here's your skin, right? So that's your skin, epidermis and dermis, and you have a hair follicle. So that's this area right here. That's a hair follicle. And you will find active stem cells and progenitor cells that are associated with your epidermis and hair follicle. That's why your hair is growing your entire life, right? So here's another example of epithelial stem cells called gut stem cells found in the small intestine. And again, they then differentiate into, again, so my computer today is really, really slow. So I apologize, I need to exit my presentation. So those committed progenitor cells then will differentiate into Paneth cells, goblet cells, endocrine cells, columnar cells. So those are specialized cells that found in your GI tract. Advantages of adult stem cells. Now, adult stem cells from bone marrow and umbilical cords appear to be as flexible as embryonic type, right? So that's advantage. They are somewhat specialized. That's why inducement may be simpler. Remember, it was pretty difficult to induce embryonic stem cells and make them to differentiate evenly. However, with adult stem cells, because they are somewhat specialized, it might be simpler. They are not immunogenic. Recipients who receive the products of their own stem cells will not experience immune rejection. So if you have a patient who need organ replacement, and we will form this organ from embryonic stem cells, that's going to be organ from another person. If you can make organ from this patient own stem cells, then it will be no immune rejection. So this tells us that some adult stem cells are easy to harvest. So it's a relative ease of procurement. So procurement, that's a big word. That means that how you collect them, that's pretty easy. They are non-tumorigenic, so they do not form tumors and they do not induce tumors, tend not to form tumors. And of course, no harm done to the donor. So those are advantages. However, we still have disadvantages. It's a limited quantity, can sometimes be difficult to obtain in the large numbers. They are finite. That means they may not live as long as embryonic stem cells in culture, and they are less flexible. They may be more difficult to reprogram to form other tissue type. Okay, so why are adult stem cells preferable to embryonic stem cells? Adult stem cells are naturally exist in our bodies and they provide a natural repair mechanism for many tissues. They belong to the microenvironment of an adult body, while embryonic stem cells belong in the environment of the early embryo, where they tend to cause tumors and immune system reactions. So another type of stem cells are induced pluripotent stem cells. So how do we get induced pluripotent stem cells? Cells from the body that will be genetically reprogrammed. So that means we add certain genes to the cell. And when we add these genes, we produce induced pluripotent stem cells or iPS cells. They behave like an embryonic stem cell. Then we will culture iPS in the lab, and they will differentiate into all possible types of specialized cells. Well, this looks like a really good deal, right? So if we can take our cells from our body, if we can add specific genes and we can make now stem cells, we can cultivate it, grow all the tissues and organs. So that sounds really, really good, because you will have a large amount of them, of these cells. You don't need to use embryo. Those going to be your own cells, so it will not be immune response. So yeah, as we said, no need for embryos. Induced pluripotent stem cells are pluripotent stem cells generated from adult cells by reprogramming. iPSCs have the same properties as embryonic stem cells and therefore self-renew and can differentiate into all cell types of the body except for cells in extra-embryonic tissue, such as placenta. No, okay, maybe we don't need this placenta, right? So here's a cell from the body, from skin, that will be induced into pluripotent stem cell. And then it can grow into cartilage or neural tissue or muscle tissue. It can differentiate. It was reported that only 4 transcription factors. So now, how do we induce them? Remember, we said we need to add specific genes. Now, how many? What specific genes? So actually, we only need 4 transcription factors that have this name, OCT4, SOX2, right, and so on. So it was reported that only these 4 factors were required to reprogram mouse fibroblast and fibroblast found in skin, cells found in skin and other connective tissue, to an embryonic stem cells. So we'll take cells of skin, we add those factors, and we reprogram them in embryonic stem cell. And we will force them to express genes important for maintaining and defining properties of embryonic stem cells. Right, so it seems like, okay, so we know now that we need these particular factors. We need to insert them into, maybe skin cell, right, fibroblast, and they can become induced pluripotent stem cells. Now, 4 key genes, these 4 key genes that we need are transported into fibroblasts by retroviruses. So retroviruses can carry target DNA that is inserted into a host cell's genome upon injection, making them ideal for incorporating the 4 genes into target cells. So here's how we create iPSCs. So here we have cells, packaging cells produce the virus. The virus is modified with 4 different genes. Right, so we have cells. Now we have virus that reproduce inside those cells. Virus is modified with 4 genes that we need. The genes and the virus then mix together, and then cells such as human skin cells are they infected with this virus, right? So now, we have a virus. Virus will carry these 4 genes that we need. Then we infect human cell with this virus, and virus will insert those genes into human DNA. So here we see 8-9 day post-injection colonies of cells with similar properties to embryonic stem cells can be identified, and colonies further magnified and shown in the next picture, right? So to create this induced pluripotent stem cells, we actually need to use retroviruses because they're going to be like a carrier mechanism that carry genes that we need into human cells, right? So here's where our advantages and disadvantages came from. So advantages: generation of iPS cells does not require embryonic tissue. So good. They can generate an autogenic, patient-specific pluripotent stem cells. Autogenic means your immune system not gonna reject it. It reduces the potential for rejection when implanted into patients, and creation of disease-specific lines allows for generation of primary cell lines for drug discovery and basic research. So we can make those cells and we can test drugs and other research using those cells. However, as always, we have disadvantages. Safety concerns due to use of virus to reprogram cells. Safety concerns due to use of genes to reprogram cells. So we changing genes, right? So how that can affect our cells, and high cost associated with generation and characterization of each iPS cell line. And they, pretty expensive in a production. Okay, and this is where I'm gonna stop for this first part of this lecture, because it's getting a little bit longer and I still have several topics to cover. Right, so I just want to remind you that in this lecture, we talked about stem cells, where we can harvest them, how they different from other stem cells, how they different from each other, and their advantages and disadvantages. Okay, so I will record the second video to finish up this chapter. Thank you for watching, and I hope it was helpful.